So I'm going to talk about managing microbes and AMF in cropping systems, some of their benefits, why you should care about them. And I want you to forgive me if I refer to you as a farmer. When I worked for the USDA, they told me, they're producers, don't call them farmers. And I didn't really like that name. Because, oh, that's loud. Because produce made me think of farmers as cucumbers. It's like, I want to think of a person. That's why I say farmer. So please take that as a compliment that I'm thinking of you. And when I first started working with farmers, of course, might have been a little dated in my mental image of the farmer. But after years at the USDA, things changed, and my view got better. So, to understand mycorrhizal fungi, you need to understand a little terminology. And what we've got here is there are arbuscular mycorrhiza. There are many kinds of mycorrhiza. There's actually 7 different kinds. The arbuscular kind are the only kind you really need to concern yourself with. These are the ones that colonize most of our crops, except for canola. It won't colonize the brassica family, but virtually everything else it's going to colonize. All our small grains are colonized by these guys, arbuscular mycorrhizal fungi. They do not make any visible fruit bodies like mushrooms. They're microscopic and can only be seen under a microscope. The word arbuscular comes from the word arbor, which is Latin. What do we do on Arbor Day? It's Latin for tree. We plant a tree on Arbor Day. So I took this picture. You're looking at a corn root, and this is one cell. See the cell wall here? And inside that cell, you can see a little structure that resembles a tiny tree. So that's how they got their name. Minuscule means tiny. Arbuscular means tiny tree. And it's an organ that this fungi grows inside of the plant roots. And it's not just in the roots, it's inside the cells of the roots. So it's an integrated part of the plant's physiology. So what happens when a seedling grows is it sends out a chemical signal that says, I want to be colonized. It sends out hormones called strigolactones. Those hormones stimulate the spores to germinate, and they grow towards the root. The plant then prepares a channel and a path through the root system to the cells that it's prepared for colonization. So the hyphae travel through there, and in those prepared cells, it forms this organ called an arbuscule. The plant, and you can't see this under a light microscope, then forms a membrane that completely surrounds that arbuscule. This becomes the interface of chemical communication and nutrient exchange between the plant and the fungus. The other types of mycorrhiza that you might be familiar with are called ectomycorrhizae, and many of them produce mushrooms. They're visible often to the naked eye if you get enough of them together as little white fibers that resemble root hairs. These do not colonize most of our plants, though. It's the endos, which is a generic term for arbuscular mycorrhizal fungi, that get inside the cells. These guys never get inside the cells. They'll colonize around the roots, and sometimes they'll get in between some of the cells around the tips, but they don't get inside the cells the way these guys do. They don't get in there and penetrate and go all through the roots. So a densely colonized roots can be 80 to 90% colonized. I've seen it myself, measured it. That's a protected root, because once the mycorrhiza that you're in there, really nothing else can get in and occupy the same space. And I'll go into that in a little more detail as we go on. So the words that you should focus on when you're thinking about mycorrhizae are arbuscular and endo. Those are the kind that matter to you. 90% of all plant families associate with arbuscular mycorrhizal fungi, which I'm going to call AMF for short from here out, because that's kind of a mouthful. That's most of our crops, and it's all of our small grains, except for the brassicas. And I'm going to give you 16 reasons you should care about AMF. They're going to increase your soil fertility. They play a major role in forming soil aggregates. They build the structure of our soil, and this increases the water holding capacity of our soil, because that's where the pore spaces in soil come from, the aggregation of soil. They produce a more nutritious product. They increase nutrient uptake, including micronutrients. They replace harmful chemicals, because if you have a healthy system, you don't really have to come in with a lot of chemicals, and you need less fertilizer. So that reduces your leaching and runoff. They also induce plants to use their nutrients more efficiently. So nutrient use efficiency is increased, particularly for phosphorus, which means with less phosphorus, you get the same yields and higher nutrient density in your final product, both. They can increase essential oil production. This is going to be really important for the hemp industry because CBD and medical marijuana have tremendous benefits in fighting cancer and other diseases. So if you can double your production by adding these guys, you've just doubled the value of your crop.
Now you said to me something last night that like industrial hemp, do plants have, I don't want to get ahead of you, but you talked about hemp here. Does hemp have a different mycorrhizae than would like a corn plant?
Yes. So mycorrhizal fungi are crop specific. There are groups of mycorrhizal fungi that associate only with legumes. There are different groups and different species that associate only with grasses. So they're particular in that particular way. There are certain species that only want to go with this one species of plant. So there's a great deal of variation amongst the different groups of AMF and the different genera and how they behave. And in the field, you really have to have a crop-specific inoculant to target that crop, or just increase your diversity and maintain some diverse cover crops to keep that stuff going in your field. And I'll go into more details on that when I talk a little bit about cover crops. They offer protection from nematodes. Boy, when I was a student, that surprised me when I read that. I said, oh, come on, it's an animal. What's a fungus going to do against an animal? It's going to just gobble it up. I'm going to share that with you later here. Protection from fungal and bacterial diseases, drought tolerance, salinity resistance, earlier flowering, more flowers, more fruit. That means more biomass and increased yields. They sequester carbon in the soil in the form of glycoproteins. So we're trying to get carbon dioxide out of the atmosphere and into the soil, where it does good, and in the atmosphere it's doing harm. Well, 30% of the carbon that a plant takes in isn't just fed to the microbes. Most of it goes directly to AMF without competition from other microbes, from inside the roots. That's how important the symbiosis is to the plants. That's like me saying to you, can I have 30% of your paycheck every month? I promise I'll do something good with it. Come on. Somebody say sure. Come on. No takers? But if it was important enough that you would die without my help, you'd give it to me, wouldn't you? That's why this relationship evolved and is so strong. Because although the plants may not die necessarily without them, their chance of survival as a species is severely limited without them. Because these guys reduce stress. And almost every form of stress that a plant can experience, these guys are actively trying to offset because that's their host plant. They cannot live without the plant. If the plant dies, they die. So they defend that plant. And pollinators prefer mycorrhizal plants. That means if I take a group of plants and put them in pots, and half of them get mycorrhizing and half of them don't, and I mix them all up, and then I sit there and count which plants the pollinators go to, they preferentially will seek out the mycorrhizal plants. No one has positively, to my knowledge, identified why or how, but insects are very sensitive to chemicals. They communicate with chemicals. They're a chemical organism that functions on detecting and sensing chemicals. And I'm guessing they're sensing the higher nutrient content. This is better for me. It's like smelling a good food. Yes, sirree, it'll grow hair, fix your teeth, and make you taller. And there's nothing these guys don't do. So this begs the question, how real is this? Can one organism really, realistically, do all those things? And the answer is no. One organism cannot do all those things. It's the community of AMF that convey multiple benefits to plants. There are many, many species. And these guys all, to some extent, will do everything on that list. But some of them are really great at some of those things. And that's what makes them desirable. And when you increase your diversity, you maximize all of those benefits. So to get microbial diversity in your soil, you have to have plant diversity. Above ground diversity literally begets below ground diversity. And I'm going to talk all about diversity tomorrow and explain how that system works. AMF affect every aspect of plant physiology that has ever been investigated. That's how integrated they are into the physiology of the plant. A large percentage of the plant's genome is dedicated to monitoring and controlling the symbiosis. For instance, if a plant is colonized and those hyphae, the filaments of the fungus that go out into the soil and collect nutrients, are not giving the plant enough phosphorus, the plant says, hey, you're not working hard enough, and dissolves the arbuscule. Those hyphae might be in an area that there's not much soil, because nutrients in soil are not homogeneously distributed. They're in pockets. And the roots go out and search for those pockets. And when they reach the pockets, more roots grow into those pockets to maximize the uptake of nutrients. So the areas that aren't finding nutrients shouldn't be getting a lot of the plant's resources. So it says, I don't need you, I don't need you, I don't need you. So these arbuscules are fleeting. They only last about a week or so till the nutrients are depleted in that area, and then new ones find new sources of nutrients and uptake them. They are a keystone species in soil. They found out if you go to a soil and you filter out everything, all the life, you sterilize the soil, and you put back all the microbes except the AMF, they will decline dramatically, because the AMF are actually helping the plants find, the other microbes find sugar and find a path to the plant. So imagine in your mind that you're the size of a bacterium. You're 1 micrometer in size. That's pretty small. And you have to get from here to that root. And you're close enough to signal the chemicals that tell you the plant is near and you want to get there. But you got all these pore spaces in soil. So how are you going to get to the plant? You got this path like this. You'll die of old age for a microbe. They don't live that long before you reach the roots. So what do? They hop on the hyphal superhighway. That's a straight shot if you can travel along that hyphae directly to the plant's roots. The plants are secreting sugars not just directly into the AMF, but through the roots also. And the AMF also secrete enzymes and other chemicals as well as sugars, so that their roots and the hyphae are both completely colonized by beneficial microbes. And as they live and die and do their things, their nutrients are captured. And that's one of the ways that nutrient uptake is enhanced. So this makes them essential to a sustainable agricultural system. And without them, you lose all that surface area, 10 to 100 times more surface area added to the plant's root system because of the mycorrhizae. And all the bacteria that would have been living on those hyphae now are no longer part of the picture. So these are all the minerals, well, most of them, that a plant requires to thrive. The ones that you normally would hear about. Silica could be on there. Nickel could be on there. There's a few that aren't normally appearing on these charts. But that's the basics. So I've already mentioned phosphorus. Guess what other minerals that the AMF can uptake? Place your bets. All of them. Except molybdenum. And they probably uptake molybdenum, but I never found a scientific study where anyone investigated it. So I couldn't put it up there. But I'd be surprised to find that they couldn't pick that up. So they're providing all of the nutrients that a plant needs. Now, let's take a closer look. I gave you a list of 16, but in an hour I can't go over the mechanisms of all 16. Yes?
On the back of that page, you have those micronutrients listed in 3 columns. A third column is
Tertiary?
Those are,
Micronutrients.
yeah, they're the ones that you need very little, right?
The trace minerals, yes.
So if you're really short of just one of those trace minerals, how does that affect the plant?
Well, plants chemically will exhibit stress. And if they don't have something they need, they will exhibit stress for that. I mean, you can see it when it's really bad. You'll see symptoms of nutrient deficiencies. But the mycorrhizae determine and detect those deficiencies chemically through the plant's inherent system of communication. And they will start trying to take up whatever the plant needs. So they try to compensate for deficiencies.
Yeah, those trace elements, as I said earlier, are the ones that are the keys to activate enzyme activity in the plant, and I'm sure probably interact with the mycorrhizae in the same way, and enzymes are needed to make things, happen. I mean,
Yes.
And so that's why they're so critical. They don't need a lot of them, but they're very critical, because without them, you can't have a good operation going on in that plant.
That's right. And although you need them in very, very small quantities, without it, certain processes just can't happen. And the plant's biology breaks down. And then the plant exhibits symptoms that you can visually see the longer it goes and the more chronic the problem becomes.
That micromix you got back there, Jim, I cut that stuff in there.
Yeah,
No,
Yeah.
you got that better micromix back here?
right. Micromix is a cocktail of really all the trace elements. It's the same we got in the Herbalife Plus that you use.
Well, I just grabbed your bag when I tank read.
Yeah.
But what's
But
that
you can
cost?
use- you can use
What's
the-
something like that cost an acre?
It's, Dennis, what is micronutrient mix per acre? It's something like what?
1.8 ounces per acre is about 678. I just looked at it.
You can use it in the starter, or you can use foliar or both. But just like Molly is very critical for nitrogen efficiency. Right, Dennis? And for what, $2 an acre, you can put it in. You don't want to use more than 6 ounces per acre annually. You can split it, either you can put it in the starter or foliar, or you can split it. But that's critical, because Dennis, most soils are deficient. Yeah.
Also, consider, for instance, scurvy. Everyone's heard of scurvy, right? What is scurvy? What causes it? Vitamin C. Vitamin C. Remember, the sailors didn't have any fruit, so they'd get a disease. So often when you see disease, it may not be something attacking your plant. There may be microbes involved in that disease in that they're opportunistic. But if your plant doesn't have the proper nutrition, not only will it present as having a disease, but disease-causing organisms will see an opportunity and jump in there, and disease will spread because of the nutrient deficiency. So the foundation of healthy plants begins right here with your soil and your nutrients and your microbes.
My theory is just about all major diseases are led by lack of proper nutrition.
I'd be surprised if you were wrong. It begins with healthy plants. Healthy plants are just more resistant to the insults of nature, whatever they are. If you have disease, you should be evaluating your system and how it's out of whack that one thing could take over. And tomorrow I'll go more into that when I discuss the benefits of diversity. So we're going to take a look. Out of that list of 16, I picked out these 4 as probably the most important and most relevant to farmers, is increasing yields, getting free phosphorus, drought tolerance, and biocontrol. So I'll start with yields. So these guys, or gals, Leckberg and Coity, they examined almost 300 experiments. We call this a meta-study, where you go and you say, I'm going to get all the experiments in this category, the category of AMF benefits. And I'm going to look at the results of all of those, and I'm going to compile that data, and I'm going to use special statistics because the experimental details are different between the studies. This guy was in the greenhouse, this guy was in the field, this guy fertilized, this guy used cover crops, he tilled, he didn't. So they looked at them all and combined them into a statistical model, and they said, in general, they found that root colonization resulted in a yield increase of 23% across all management practices. The good, the bad, the ugly. So you can imagine that the good was a lot better.
But did they quantify quality? Because that could be another attribute.
Not in this study.
Yeah.
But they looked at that, and then they found that, AM fungi, root colonization, and percent root colonization have a direct impact on your yields. So AM fungi make you money by increasing your yields. Free phosphorus. Phosphorus is the fulcrum of this relationship. In nature, phosphorus is scarce, and plants use and receive very little of it. It's more like a secondary nutrient. Even a tertiary nutrient can provide enough phosphorus to keep a plant alive long enough to reproduce. So because it's so scarce in nature and there's so little available, this relationship evolved, and the chemical decision that plants make to determine how much sugar, how much carbon should I shunt to my mycorrhizal fungi is directly determined by the amount of phosphorus that it can uptake. And if we flood the system with phosphorus, the plant says, I don't need mycorrhizae. And then you can just tick all those other benefits off the list because the plant doesn't measure all those other benefits. The decision is made based on phosphorus. So we have to be really careful in how we manage our phosphorus. And I chose this study, even though it's an older study, I think it's 1979, because this is a very typical response to colonization. Yes?
Is there a threshold where your phosphorus, what's the target number for too high phosphorus?
Depends on the form of the phosphorus, how available it is, so there's no magic number. But in general, and this is according to Ray Archuleta, that the cutoff is 200 parts per million. However, I had someone with much higher phosphorus than that say, we've got a problem with this field. We did everything the same, except on one side of the field we terminated our cover crops a little earlier. We even used the same herbicide to burn down the cover crops. And we can look at the field and see that this side of the field is doing great. They planted corn after their cover crops, and that side of the field sucks. It looks terrible. We don't think that there's any mycorrhiza in the field, but it was the only thing we could think of that might be affected by a slightly early termination of the cover crops. So she said, would you look at the roots? And she sent me root samples. She didn't tell me which was which. So I just cleared and stained the roots and scored them and sent them back to her. Said, here's your results. And she said, wow. So what I found was that where they had the early termination of the cover crops, zero colonization, which is what she expected everywhere because of the high phosphorus levels. But what I found was in spite of the high phosphorus levels, the other side of the field still had low colonization. It wasn't very high, but it was something. It was below 25%, which we consider on the low end. The stuff from the bad side of the field got the double whammy. Not only was it denied the benefits of AMF, but it was colonized by a pathogenic fungus. The other side of the field that had the mycorrhizae had no pathogenic fungus in it because they don't share. They don't play well with others. The pathogen couldn't penetrate the roots, even with only 20% colonization. I was surprised that that was enough, but it was. And they were well above the 200 part per million mark on their phosphorus. So it just depends on a lot of different factors that influence the system. I recommend that plants be slightly hungry, that it be a little lower than their normal need, somewhere between 100 and 200 and closer to the 100 mark, if you don't absolutely need it. Because those tests that we give don't really measure how much phosphorus is in the soil. It measures how much phosphorus that test can detect. Think of all those microbes. What, 2 elephants worth or something per acre? Think of all those microbes, and every one of those cells contains DNA. And DNA is made of phosphorus. None of that gets measured. There's all sorts of phosphorus that's bound up to different elements that doesn't get measured. They don't measure what's bound to organic matter, for instance, which is really important in holding onto that phosphorus and letting it go when it's time, when the plant demands it, because when the plant demands it, the microbes go to work to make it available. There are some forms of phosphorus, for instance.
You don't measure it because it's not in the inorganic form?
There's too many forms. There's too many forms for them to measure it all. It would take 100 tests because it binds to so many things.
Does that relate back to our manure that we talked about earlier, that we're not detecting, where we buy augment manure in the analytical?
It could tie into that, yes, because we don't detect it all. We only detect what the test is capable of detecting, what's separated by this type of solvent or that type of solvent. And that's basically 2 tests, right, for what's in acid soil and what's in alkaline soil. But there's a lot more there, but we don't know how much it is. But it's always there. It's a matter of freeing it up. But if you don't have organic matter, you're not going to trap very much of it. It's going to be lost. So this is a very typical response. In this case, we've got 40-day-old soybean plants. Look at the dry weight of the plant, about a third more. That's about a 30% increase in biomass. Copper, negligible. Copper is toxic to most fungi, so I wouldn't expect a big throw there. Zinc, tripled. Phosphorus, doubled. Microbes provide 50% of the phosphorus that your plants need. When they're gone, you've crippled the ability of your plant to uptake nutrients and the rate at which it can uptake nutrients. And this is how it works. The plants, through photosynthesis, create sugar. It's glucose, not sucrose, but this is just an example, okay? So they create sugar, and they feed sugar to the microbes, and in particular to the AMF, and in return, the AMF produce all those nutrients, including phosphorus, which, by the way, have you guys noticed in the last decade how phosphorate prices have gone up and up and up? They're never going to come down. They might trend, they might vacillate up and down, but the trend is always going to be up because we're going to run out by 2040. By 2040, we're going to be dependent on foreign sources of phosphorus for our food security in this country. What do you think will happen to the price of phosphorus then? It's like oil. In fact, phosphorus prices, food prices, and oil prices all track now and have been for many years, ever since it was discovered that we're almost out. And then we'll be dependent on who? Morocco has 70% of the world's phosphate supply. Hope we're on good terms with them. Yes?
So how much phosphorus do you have to have for the microbes to work on?
Just try and keep it from being too high. So if you don't go over the 200 parts per million mark, you're not going to kill everything, but if you're below that, it's better. But everybody's plant and soil is a little different, and they have a little bit of different soil history and different levels of organic matter. So how much you need to get into your plant is going to be a little different than other people's. So you know from your experience what works best on your farm, but you should soil test religiously.
But I mean, you have to have some. You can't just produce it out of thin air.
Yes, you have to have some. I'm just saying keep your parts per million rates below 200 and better closer to 100. And that's pretty high. That's plenty for almost every crop out there. Potatoes might want more, but they're a real high phosphate demand.
It also depends on how healthy your system is and if the other elements are there to complement the phosphorus, because if your system's out of balance, a lot of that phosphorus is going to be tied up that you put out there. Okay?
Yes. It's just like you need a nutrient, one nutrient to mobilize another nutrient. So you have to have the balance. You have to have the healthy soil. Phosphorus isn't the only thing that's essential to healthy soil. I'm focusing on it just in this relationship with AMF. So the AMF then produce the nutrients, and the plant uptakes those nutrients. It's literally a budget. And the AMF can be attached to more than one plant. And there's competition for this sugar between the AMF. So the more AMF diversity you have, the more competition you have, the harder the AMF worked and the lower the cost of phosphate going into your plant. That's why diversity really plays a key role here. But what do? We come in with a truckload of phosphorus fertilizer, expensive phosphorus fertilizer, and we dump it in the system. And then the plant says, oh, I've got enough phosphorus. I don't need to give you any sugar. And since the mycorrhizal fungi can't live without a host, if their spores have already germinated, they then die because there's not enough food. And you can say goodbye to your free phosphorus because you have to keep trying to do the job of the fungi now. Less than 20% of the phosphorus you put into your field will get into the plant. That's because it's like a magnet. It attaches to everything, and the minute it hits the soil, it's becoming unavailable. It's binding to things. And only the microbes are going to make it available. The plants can uptake some without microbes, but not very much and not very efficiently. That's why they love microbes. That's why plants literally grow microbes. They shunt sugar into the soil to propagate microbes. They're farming. The plants are farming. We're farming the plants.
Calcium phosphate.
There you
So
go.
there's a good example of what happens in your soil. Calcium, phosphorus has negative charges, calcium is positive, so you create this. The plant can't access that, that's insoluble. And believe it or not, I worked on a hog farm out in South Dakota for a big operation, and they were losing pig capacity. So I went in there, I treated for a year, I had that lagoon just a humming. It was even pink. I had the purple sulfur bacterium. I thought, I'm home on this one. They're going to gain a lot of capacity. And they pumped it out and said, Jim, we only gained about 20%. And I went out there and I burned out all the organic matter out of there, brought it up, and it was lined with calcium phosphate, just like that. All that material had passed through all their pigs for years and into that lagoon and dumped. And it takes years for microbes to break that apart. It doesn't happen overnight, but they do. But I mean, that was totally unavailable. And that's a good example of that.
So here we go. The phosphate is being bound, so it becomes unavailable. And if you have a lovely truck like this, and let's say you bought 10 gallons of gas and put it in your truck, and then use the other 40 gallons to kill fish, well, that's how we treat our phosphorus. It erodes away, it runs away, it leaches away because it's not getting into the plant. But it is getting into our water systems. We call this eutrophication, but I have another word for it. I call it wasting your money. That's like throwing away your fuel, but paying for all of it. So AM fungi save money on chemical fertilizers. And by reducing our chemical inputs, we increase not just the AMF, but all the other microbes that live in the soil increase too. Think of the billions of things that live in soil. You can't manage for a billion things, but if you manage for these guys, for the AMF, everything else comes along because everything kind of needs the same system. Drought tolerance. Now, when I was a student and I read about this, I said, really? These hyphae are really small. They're smaller than root hairs. It's like big straw, little straw. How much can you suck up, you know? How much water can they really uptake? And Ruiz, Lozano, and Ascon came up with this really clever experiment. They asked themselves the same question. They said, we're going to figure it out. And what they did was they got a tube, and they used sand as their media, and they put a 50-micron mesh between these compartments. The roots are too coarse to fit through that mesh, but the hyphae are small enough to go through it. Then they injected water into the bottom to see how much water the hyphae could deliver directly to the plant. And that was the only water source. But then they said to themselves, mycorrhizal fungi are so good at uptaking and finding phosphorus and developing and delivering it to the plant. How do we know if the plant performs better that it's not because of that phosphorus? How do we know it's really the water? So they gave their control a fertilizer, a pea-fertilized control. And then they tested this with Glomus fasciculatum and Glomus deserticola. What does this word remind you of, deserticola? Desert. A desert. That's where it came from. Who do you think is the winner? The phosphate fertilized control, deserticola, or fasciculatum? Place your bets. That was deserticola. So 215% more fresh weight than the fertilized control, 150% more fresh weight than the fertilized control. That's a lot of water. That's more than double that it could have uptaken on its own. Because of the tremendous surface area that is available to uptake nutrients and water when you have all those hyphal filaments out there in the soil. They become an extension of the root system. Furthermore, they can get into pore spaces too small for roots and even root hairs. And those really small pore spaces have higher surface tension. They're harder to extract the water from it, and they can get into those pore spaces and make that water available too. And that can really save your butt in a drought. So money saved and losses due to drought. I'll tell you a little story. When I was working for the USDA, I had some plants. The whole farm was pretty much planted to corn, and I was doing a cover crop experiment, and we were looking at using cover crops to select the most beneficial microbial community for the cash crop. So we had different communities of cover crops, and we'd harvest them, and then we put in corn. And it was, 7, 8 years ago when we had that massive drought hit the Midwest. And one day the farm manager came and said, Wendy, you better go look at your plants. Now, I was going to harvest those plants at 30 days because I was going to just process the roots and measure the mycorrhizae in them. And when he said that, I thought they were dead for sure, because my plants weren't being fertilized. I had told him, don't bother fertilizing them. I'm going to dig them up and chop up the roots anyways. So everybody was making fun of them because we had the sea of emerald green plants, and there's Wendy's section with all these lighter green plants. They really stood out because they were nitrogen deficient. So came time to go out there, and I'm walking through this, and we're right in the middle of that drought, and all these emerald green plants, their leaves are all curling closed. They're trying to protect themselves from the sun and the heat and evapotranspiration. I get out to my plants, and the leaves are wide open. Although they did kind of wave at me and say, hey, we could use some nitrogen over here. But they showed no sign of drought stress. And that was just amazing to me. If I had known that drought was coming, I would have done a big drought test experiment. Yes?
So which cover crops were best suited for the program?
The mixes with the highest diversity won. Biocontrol agents. AMF protect plants from a broad range of plant pathogens. By the way, I did some work last week on asking myself, how many pathogens are there in the world that affect plants? Does anyone know what that number is? I thought it would be pretty low. I used to think there aren't that many pathogens compared to the billions of known beneficial bacteria and fungi. There were 75,000 known plant diseases. And they don't all affect the same plants. Some are unique to certain plants. And they're not all caused by different organisms. Some are caused by the same organisms. But it's still a drop in the water compared to how many different species of organisms are in our soil. It's less than 1%. So here we go. We've got anthracnose disease, root rot in legumes, root rot in other things, fusarium wilt, Panama disease, take-all disease, root rot disease complex. These guys that cause this, these complexes, these are nematodes. They're opportunistic microbes can get into a plant after the nematodes have breached its primary defense, which is its outer skin, just like us. My skin is not going to just randomly get an infection, but if I get a cut, I'm more likely to get an infection where that cut is. Well, the same thing happens to plants. So they damage the root, and then everybody else moves in for whatever they can get. We've got damping off, stem root rot, verticillium wilt, and this is not the full list. This is just a little subset. I said, I'm only going to put on here what I can get on one slide, so I just picked the most common diseases. How does that work? And in particular, what really fascinated me was how does it work with nematodes? So here we are. These colored balls represent different species of AMF. So the orange guys are the same species, the pink guys are the same species. They can interconnect plants, even if the plants are different species. They can also connect to each other's hyphae if they're the same species. So this orange guy can connect his hyphae to that plant indirectly through the hyphae on the other plant and fuse them. They call it anastomosis. So here comes our nematode, and he's hungry, and he starts chomping on this plant right here. And then the AMF, which extend beyond the roots, because they're literally an extension of that root system, go, oh my God, this guy's going to kill my host. And they take over the plant's defense mechanism and send a signal that says, turn on all your chemical defenses against nematodes. And it's activated directly by the AMF. And because they're interconnected to surrounding plants, the signal goes through that hyphal network just like the internet and reaches the surrounding plants. This plant still gets chomped on before this kicks in. But now the next guy that comes along, or if this is a migrating nematode and wants to go to another plant, the next plant already has its defenses, which makes it distasteful. So he's not going to chew that one. He's not going to chew the next one. Or the next. He's got to go to a naive plant that hasn't been warned he's coming. And in this manner, the amount of nematode damage is suppressed because it can't get all the plants. So what does the science tell us about our superfungi versus nematodes? AMF versus the soybean cyst nematode. Damage offset 36%. In a review of nematode and AMF interactions, the AMF were most effective against the root knot and cyst nematodes, with plant damage reduced an average of 22%. They're particularly effective against Telenchorhynchus, the stunt nematode, where they caused a 21% decrease in nematode numbers. They were killing the nematodes. Now, I was like, what? This has got to be a chemical thing, right? But what was really amazing was they were killing female nematodes selectively. Selectively killing female nematodes. I'm a biologist, so I know if I want to control a population, what I do is I go out and shoot all the females. That's how it's done. That's what biologists do when they need to control a population. So this is kind of amazing. Versus migrating nematodes, mycorrhizal plants were more than twice the size of non-mycorrhizal plants. So, oh, I'm missing my you save money in nematicides and other interventions slide. So you're going to save money when you don't have crop losses and you don't have yield losses due to nematodes. Must have accidentally deleted that one when I put in my tillage slide. And is there anyone here who's never seen something like this happen? Who's never seen that? So we've all seen this. So I pose a question to you. Is this where your soil belongs? This is not just your soil, it's your topsoil. It's the richest, most important, most biologically active, most nutrient-dense part of your soil that you are saying goodbye to as your neighbor collects it or whoever's downstream when it rains, or wherever the wind deposits it. It's your best soil. That's the number one problem with tillage, is it causes soil erosion. But what does it do to your microbes? So here's a seed, and this is our soil, and we planted it. And when I worked for the USDA, I discovered pink seeds because we put so much toxins on them. And I had someone ask me, well, what mycorrhizal product should we carry in our store? And I said, I don't know, are your seeds coated with fungicide? And he just said, oh. Next time I went to the store, you know what they're selling? Fungicide-coated seeds and fungi. So, your plant grows, and these little colored lines are your mycorrhizal hyphae germinating and colonizing that plant. But we never plant just one seed, we plant a bunch of them. Roots interact as the plant matures, the hyphae and the fungi interact, and then we harvest. And this is what's left in the soil, all these hyphae. I know those are hard to make out in this slide. But
Is that an actual slide, picture of hyphae?
No, it's a diagram
Okay.
to make a point. Can you turn off the light back there for a minute so people can see this? We'll turn it back on in a minute. Because this is hard to see.
Dennis knows where it's at.
There we go. So here's what's left. We've got all of these hyphae left in the soil. And then what do? We plant again, right? And then the plant germinates, and it comes in contact with hyphae and spores and sends out signals, and it gets colonized. And that's what's left if you remove everything that's not in direct contact with that plant. And I just randomly drew it and then copied and pasted, but it's to make a point. That's how much of all that network that the new seedling immediately has access to. Let's compare that to a non-colonized seedling. What does that guy have? Look at the difference in the volume of soil. This is one of the important ways that AMF increase the uptake of nutrients and waters to plants, is they increase the volume of soil that can be explored for access to those nutrients. Now, in many crops, and most of my experiences with corn, the amount of phosphorus that you get into your seedling in corn before the 5-leaf stage, determines your yield potential. All the phosphorus you add after that is burning your money. That decision has been biologically, biochemically determined by the 5-leaf stage. And it's based on how much phosphorus directly is in your tissue, is in the plant's tissue. So now we're going to till. We chop all that up. So these lines represent breakage points. So I'm going to take away everything that's not directly connected to the plant if we had tilled the soil. Then the plant would have just been colonized by these little fragments. That network wouldn't have been there. Because when spores germinate, they're still attached to that network. And they send hyphae out to the plant, and then they race through that network. And they can absorb the damage areas, and they can bridge other places, and they use that network to move quickly without spending a lot of energy. It's a difference between going down a deer trail and hacking a path through the jungle with a machete in the amount of energy expended to colonize that soil and get out there. And that means year after year, these networks expand and grow and get bigger and denser, which is great news for you. The benefits don't plateau until 13 to 17 years. One tillage event sets it to 0. Start over. So we just shot ourselves in the foot as far as our yield potential, because this plant is not going to be able to uptake much more phosphorus than this plant. But when those hyphae find that there's no network to connect to, what are they going to do? They start growing a new network. Who provides all the sugar to this fungus? It gets it all, 100% of its nutrients from the plant. You just increased the cost of the symbiosis. Carbon that could have been used in other ways to increase your other microbes, to increase your growth, to increase your bricks, is now being spent to rebuild this network. So how does soil respond to tillage? Well, first of all, it kills AMF, because not all the species out there can survive having their hyphae chopped up. How long would you survive if we chopped off your arms and legs? Probably not too long. AMF builds soil structure, so tillage increases erosion. It breaks down the structure of the soil. Tillage causes compaction, loss of carbon, and I'll talk a little bit about this because Don spoke on it. When you dig up the soil deeply, you introduce oxygen into areas that are primarily dominated by microbes that don't need very much oxygen. Many of those are nutrient fixers and mineralizers that manage your nutrient cycles. They're microaerobes. They only need a little bit of oxygen, and that's all they've got, and that's all they need. But what happens when you till and introduce a lot of oxygen deep into your soil, the aerobic organisms have a population boom. Suddenly, we can live here because there's more oxygen. Now, what do we inhale? What do we use from the air when we inhale? Oxygen. That's what we need. What do we exhale? All those aerobic microbes are pretty much doing the same thing. They're exhaling, secreting as a waste product carbon dioxide. That's where your carbon goes. It's burned up in that population boom and sent out as carbon dioxide. So you suffer a carbon loss because your soil organic matter is consumed to supply that carbon. It reduces the water holding capacity of your soil. It kills predatory insects because of the timing. In Mother Nature, the pests come out first, the little things that the predators eat. Because if the predators came out before their food was out, they'd starve and die and wouldn't exist. So first the pests come out, we get out there, and we till, and that kills these guys. Kills a load of beneficial organisms, including your spiders. And it plants weeds. And this is particularly important to organic farmers who use tillage to control weeds. You're trapped in a vicious cycle. So how come we dig a hole, or cut a hole, a slice in the soil, and we cover the soil, and we put the seed in there, and we cover it, and we pat it down. We even have a little patter, little thing on our tractor that tamps down the soil and keeps firm seed to soil contact. Why do we do that? Why don't we just throw the seeds out there? What's the point? Mother Nature just throws the seeds out there, right? Wind-borne seeds, animal-borne seeds caught in fur. Well, they're protected. They're protected when they're planted, when they're covered. They have access to water, which helps them start to swell and prepares them for germination. They're hidden from seed predators that could otherwise easily find them. Little mice and beetles and insects and all sorts of things want to eat those seeds. But what do? We take all those windblown weed seeds that are laying on the ground, and the temperature extremes on the ground are hard on the seeds too. That's all buffered where you're insulated in the nice cozy soil, waiting for the right moment to germinate. But we come in, we till, and we actually plant all these seeds, all these wind-borne seeds when we till. So we sustain our weed problem. Fortunately, I know a weed scientist and spent quite a bit of time with him at the USDA, and he came up with a solution. His name is Randy Anderson. You organic people should write down that name and Google him and find some of his work. And what he said was, you know what? If we go with our cover crops, cool season, and then switch to warm season, the cool season stuff outcompetes the warm season stuff. And the warm season seeds germinate and die because they're shaded out. And then the opposite happens when you switch. He says if you do that for 3 years, your weed problem will become insignificant because you use up the seed bank as long as you're not tilling. As soon as you till, you start planting weeds again. So to help you organic farmers who asked about the weeds, this is one solution. One solution. Tillage is not your friend. Tillage is sustaining your problem. But we get trapped in a cycle because we have to have a crop. And if we can't manage our weeds, what are we going to do? So you got to break that cycle. And he came up with a way to break the cycle. Yes?
You talked about even these effects from tillage, but the colony of the mycorrhiza, are you seeing less of effect with, say, fall tillage versus spring, or is it just as detrimental either or?
All tillage is detrimental. Timing can make it worse, depending on what you kill with it, but it's always going to destroy your hyphal networks. And it's, depending on the timing, also going to have a negative impact on your predatory insects.
How quickly do they actually die off after tillage?
The AMF?
Yeah.
The ones that can't survive without sporulation, without spores, are immediately destroyed, and they come back more slowly. In fact, the really large spores of Gygospora are the first ones lost and the last ones I ever see in any farm. I almost never see it. If a farmer sends me soil and he's got Gygospora in it, I call him up and say, how are you managing your farm? And he goes, what? I found Gygospora, you know? So it's a good indicator. If it's gone, because it preferentially likes to colonize perennials. And it's responsible for some of the largest, most intensive hyphal networks out there. That's what it specializes in. Remember I said they specialize? They all do a little bit of everything on that list, but some of them are really good at some things. Those guys make these massive exploration hyphal networks and take up probably more nutrients than any other group. And they're the first ones we lose. Cover crops can save them. They can't withstand having their hyphae destroyed or damaged. Small bits of damage, they're the only group that does this. They can actually heal their hyphae when they break, but not when you till. That's too much damage for them. They're instantly gone. What spores are left, if you don't till again, you might recover those numbers if you have a compatible host. But otherwise, they just disappear, and that's what I see. They're gone in most agricultural ecosystems. So keep in mind, too, that roots can't penetrate a compaction resistance of 300 psi or greater. So if you're tilling and you have a compaction problem, you need to get out there with a penetrometer and see how deep and how serious that is. Because if it's higher than that, you're going to have to till if you need to get rid of that compaction layer. If it's shallow and it's hindering your plants from getting down to where the water and the moisture is. But immediately after you till, you should come in with a really dense cover crop of deep-rooting plants, because it's going to try and recompact. That's what happens. As soon as you till, you break down your soil structure, and everything starts compacting again, every time you till. But if you get your cover crops in there before it compacts, immediately, those plants with tap roots will start creating channels. And you can make that the last time you ever have to till. Because if you've been tilling, I guarantee you probably have a compaction problem. Am I wrong? Is someone tilling saying, I don't have compaction? I've never heard anyone say, I don't have compaction and I till. It's never happened at a conference, no matter how many people were there. Okay. Some of these are particularly deep-rooting, forage radish. And you can't overdo too many of them. You got to try different combinations, see what's gonna work best for you. Rapeseed, that's canola. This is a brassica. It's non-mycorrhizal. It will not reduce your mycorrhiza by having it in there. If you just grow nothing but canola year after year, canola after canola, well, yeah, you're going to lose everything. But having it as part of a mix increases your diversity. And in an experiment we performed, we found that even though we balanced our cover crop mix by reducing the other seeds when we added canola to it, that adding canola still increased the mycorrhizal colonization rate when we followed with corn. So it's good to have this competition. Because you know what mycorrhiza say when they see a non-mycorrhizal plant? Oh, I have to work extra harder because I don't want this guy to take over. He's not going to host me. It stimulates mycorrhizae to have that kind of competition. One of the things we try and do in agriculture is remove all the competition. We have to reintroduce it because it makes things work better. But it has to be introduced in a way that doesn't harm our bottom line. And that means we control what our weed is. And in this case, that would be the canola or the cover crop, but we have control over that. We're using it for a purpose and we're generating what we need. It's not a weed anymore. So we've got rye, sun hemp. I don't know about, sun hemp is not related to the hemp plant, to cannabis. It's a legume. Asparagus, I threw this one in here. I think someone should try asparagus. Asparagus loves Gygospora. I always find it on asparagus plants. I use it in the greenhouse as a culturing plant. It's great for generating Gygospora spores. It's a perennial. Gygospora takes a little longer to actually complete its sporulation cycle because it's attached to a perennial and it expects that plant to last a little longer. So someone, I think, needs to try this. So what if you kill it after, before it goes a whole year? It doesn't have to go all that long. But I'm encouraging someone to do an experiment on their farm and just try some asparagus. See how it works. Sunflower, very, very deep taproot. Chicory, another one. A lot of people use these blends to feed deer. They'll throw in some oats and other things. Oats, by the way, is a highly mycorrhizal plant. It associates with almost every family of mycorrhiza. So it'll really, really promote your mycorrhizal diversity. If you're not asking yourself this question, you have not been paying attention. So how do we get more of these guys? And if you've never seen the spores, this is what they look like, except for that guy. That's a different fungus. But most of these are mycorrhizal spores. From this area, by the way. These are spores I collected from Minnesota soil. So the first question is, which would you rather eat? If we offered you this for lunch, would you have still come here? If this is all you ate, would you be healthy? Is that all you need? All the vitamins your body needs? All the minerals? Yes? No? Could you live like that? Not just because you like this stuff, but because you need this stuff. You need the roughage. You need the complex molecules. You need the proteins. You need stuff that you can't get in that. But this is how we feed our plants. We don't give them everything they need. We give them chemicals. And then we wonder why they're not as healthy as they should be and why they're prone to disease. Well, they're not on a healthy diet. They have to be on a healthy diet too. So let's go back to Leckford and Quady, the guys that did the nearly 300 experiments. And they found that in general, you got a yield increase of 23% across all management practices if you could increase your root colonization. So that begs the question, how do you increase your root colonization? Can we do that? Well, one reason you already know. How does the plant decide whether or not it even wants to be colonized? Anyone? Phosphorus levels, right? So one of the ways you can do it is to make sure your phosphorus levels aren't excessive. But root colonization was increased most by inoculation. 29% increase in colonization. Short and fallow, 20%. Reduced soil disturbance, 7%. Add that all up. Inoculation really means good management too, because if you don't have the management in place, you're going to kill your inoculant. When you inoculate, because these inoculants are expensive, you have to nurture your soil and maintain best practices to keep that inoculant alive, because what you want it to do is proliferate and spread throughout your field and grow in numbers. Short and fallow. Don't you dare. This is so old school, fallow. We used to think we had to rest the soil. We know now that that's not true. That was just our lack of understanding and knowledge. And today we call it ignorance. But back then it was where we were. Today we know that leaving the ground bare destroys your soil. Destroys your soil fertility and causes erosion, and you lose your topsoil. So don't ever leave your soil bare. In fact, if you can see your soil, you don't have enough covering it. There should be residue covering it or live plants covering it. You should not be able to see your soil. This equates to using cover crops. Simple as that. Fill in between those rows. And companion crops. Reduce soil disturbance, that's no-till. And after today, I hope you're thinking no-till is a no-brainer. It really is. So the unanswered question here, and I answered it already because someone asked, was how much phosphorus do we really need to maintain our yields in our AMF populations? Well, here's what we have. We've got root colonization going here and yield up over here, and here's our inorganic phosphorus levels from low to high. I didn't put numbers on it because it varies depending on the crop species and its need and the condition of your soil and how much organic matter you have. But we know that root colonization declines as the inorganic phosphorus levels increase. But we also know that plants respond to fertilizers. So the other side of the coin is our yields increase as we add our fertilizers. And this is a new breeding target for plant breeders. And I know we've got a representative of Albert Lee Seeds here because I have been wanting for a long time to get someone to breed for this target. That intersection is the sweet spot. That's where you're getting the most bang for your buck. That's where your nutrient efficiency is maximized. So you get the highest yields at the lowest cost and the minimal inputs. And we should breed our plants, in my opinion, to move this intersection up in this direction so that we elevate the yields even with lower colonization rates. That's the future of agriculture. Breeding for low input systems. The way they breed plants now, they go to places like Brazil, they seed in their plants and they put the maximum inputs. The highest inputs you can imagine. Why? Why do they do that? They can grow the plants more cheaply. They don't need to do that. They do that because they're trying to create the maximum yield potential. So they can say, if you do everything this way, you can get this much. But if you do everything that way, your profit margin is going to be crap. Sorry, that's the only way to put it. Because your input costs are going to be so high, and to bump up that little bit that little bit of yield, you're going to spend a lot of money. It's the higher you try and bump it, the more expensive it's going to become to move that line, that yield line. There's a barrier there. It's called the physiology of the plant. It can only do so much. And you've killed off all your microbes. So what they're actually doing, and this is becoming alarming because we'll never become sustainable, is some microbes become a yield drag. Some AMF, there's a group called Rhizophagus that can become a yield drag under high input situations when you have too much. So they're actually selecting now against the symbiosis. And just this last 2-3 years, I have seen corn and other crops that are incapable, new varieties that are incapable of forming a mycorrhizal symbiosis. This is rather alarming because we will never become sustainable without this symbiosis. And we're going to run out of phosphorus. So we'd better be put on alert that this is the way plant breeding has been going, and it needs to change. Not necessarily for you guys. You guys are organic, yeah? Albert Lee? Are they you guys organic?
Albert Lee Seed House?
Albert Lee Seed House? Oh, I thought the guy was here from Albert Lee.
Tomorrow morning. They're organic and non-GMO.
And non-GMO. Awesome. So profit margins per acre are more important to your sustainability than yield. When these big seed companies come out and say, oh, we're going to give a prize to whoever has the highest yield, ask that guy when you go to those talks, how come you don't get that yield every year? How come they don't? Because all the conditions have to be just right. And if they're flooding their system with nutrients to try and make that yield that little bit more, not only are they reducing their profit margin to get that little bump, but they're destroying the quality of their soil. They're never going to see it again. So don't let them sucker you into this big contest with other farmers. Just quietly collect your money and laugh your way to the bank. You know? Thank you.
Okay.